Welcome everyone to Vidyastra quick revision classes. In this video, I'm going to discuss 10 highly important topics which are surely going to come in UPSC Prelims 2025. These are a little difficult topics, but I will break them down for you. I will make them simple for you. Okay, and you can seriously expect at least 4 to 6 marks from this section. Biotechnology is always an important section, and this is the section that I'm going to cover today. So let us first start with environmental DNA analysis. UPSC has already asked question in 2023 and in 2020 also. Okay, so what is environmental DNA analysis? DNA analysis, if you need to understand, we acquire DNA from any organism and we analyze it. There are different steps to it, like sample connection, DNA extraction, amplification, sequencing, data analysis. Okay, but this is not DNA of one particular organism. This is an environmental DNA analysis, means we are collecting DNA from a complete population. It can be a tropical rainforest, it can be a sea, it can be a pond. Now why we are doing this? Because there are some problems, there are some issues that we need to solve at the population level, like identifying invasive species. If we have to identify invasive species, we cannot just go into a forest and identify each and every species. Like further, if we have to identify species diversity. Okay, now if we were to identify endangered species, if you have to track pathogens in a complete population, so all this can be done with environmental DNA analysis. Now, what is environmental DNA analysis? What we do is we collect the DNA of the complete population. Complete population. Let's say we take sample of the DNA of complete population. It can be taken from air. It can be taken from water. Let's say you take half a liter water from a pond. There will be DNA from different organisms. Why there will be DNA from different organisms in that half a liter of pond water, or let's say 1 cubic meter of air from a tropical rainforest? Why there will be a DNA? Because DNA, we continuously give DNA in our surroundings. Like when we shed skin, our skin has DNA. If we are releasing mucus, that has, our feces have a DNA, our hair has DNA, gametes has DNA, decomposes, tissues have DNA. Let us say a tree is decomposing, then there is DNA. It might be released in air or in water. So there are different species in the population. They are releasing DNA in the environment, and that DNA we are collecting and then going through different steps and trying to analyze various characteristic of that population. It can be understanding invasive species, endangered species, species diversity, anything. Okay, now these merits and demerits you can you easily understand now. It is non-invasive because, as I said, you don't have to target every individual. It detects elusive or rare species because, let's say, the number of members of one species are only 2 in a complete rainforest, or let's say 4. Okay, now it will be difficult to identify a single individual, but from a DNA analysis we can easily identify. Environmental DNA analysis. Now there is this limited reference database and degradation of eDNA in environment. What is the issue? As I said, this DNA is not extracted directly, it is extracted through indirect sources like shed skin. Okay, now but it is not like that the skin that you have shed will remain in the environment for a whole year. So it can degrade and it can be lost. So we might not be able to detect some species which are existing in that population. Okay, now then is the limited reference database. Why? What is this limited reference database? Because one thing about environmental DNA analysis is you need some data from previous time. If you have to analyze whether a species exists or not, but then you have to analyze it from the perspective of what happened earlier. You're getting what I'm trying to say? That is important. If you do not have any reference, if you have not collected any DNA previously, then it is of lesser use. Okay, now these are very important topics, a little difficult, and I will explain it. First, I will have to explain you gene expression. Now RNA, DNA, gene expression, protein synthesis, ribosomal DNA, transcriptomase, messenger RNA, siRNA, all these things you know. Okay, but I'm going to break down this for you, and please concentrate on what I am saying. You will be able to understand. Now you need to understand that what is gene. Okay, gene is gene in our body. Any gene in our body is like a scientist. He has all the information what is to be done whenever something happens in our body. Okay, RNA is like an engineer. RNA is like an engineer. This engineer takes information from the scientist and then does the other half of the work. Means it applies the knowledge that is present in DNA. Okay, you are getting me? So gene has the information, RNA applies this information, and this all this we are going to study here. Now, let's say, let me give you an example. We have a gene which knows what to do when glucose levels increase on the body. Let's say we ate two diets which had high level of glucose. Now, we all know that insulin has to be released in our blood to reduce that glucose level in our blood and store that glucose as fat. But how is this happening? Okay, first, that is not an automatic process. Means it is automatic, but there are the actors involved here. And what are those actors? These actors are the DNA and the RNA. DNA has the information what we have to do if so much glucose comes in our body, and RNA does actually does means release. It takes action. It releases proteins which leads to release of insulin, which controls the blood sugar level in the body. And everything happens like this in our body. There is a stimuli, then there is then that gene is activated, then there is gene expression. How gene expression takes place? Gene expression means that gene's information is converted into actionable outcome, actionable product. And who does this? It is done by RNA. Okay, so we are going to study all this. Now, multi-omics is the study at different levels. What we are studying is genomics, transcriptomics, proteomics, metabolomics, everything I told about. Genomics means DNA, transcriptomics mean RNA, proteinomics means protein, and metabolomics, as I said, those insulin, etc. Now, multi-omics, what it does, it studies at genomic level, transcriptomics level, proteomics level, and metabolomics level. But why are we studying? Because let's say we have to devise a drug for diabetes. Now we will need to understand all the 4 steps. So multi-omics does this. We will have to understand all the 4 steps, how the gene expression, how the means what is the information stored in the gene, how is the information transfer transcripted by DNA, how then protein synthesis take place, and how that protein synthesis leads to insulin. Okay, so multi-omics does what? Understand all these 4. Look, these components are genomics, transcriptomics, proteomics, and metabolomics. Okay, and as I said, what is it used for? For drugs, enhancing crop productivity. It can be used in other things. Wherever any complex biological process is taking place, it can be used. Okay, now but it has high computational requirements. Why? Because so much data is there. Data integration complexities, because these are different levels. Okay, now multi-omics we study. Transcriptomics I already told you. Transcriptomics about what? Omics is studying about how RNA transcribes that information which our scientist gene has. Because RNA is what? RNA is an engineer. Okay, study of complete scenario of RNA transcript produced by genome under specific conditions. The techniques can be RNA sequencing and microarrays. This is an older technique. Okay, understanding gene expression, as I told you, uncovering non-coding RNAs with regulatory roles, identifying biomarkers for diagnostics and therapeutics, developmental biology. Let me first cover this topic, because it is a bigger topic and you will be able to understand genetic markers in conservation biology. Now, this is let us first do that only. So we were here. Transcriptomics. Okay, so we studied about multi-omics. We studied how different things take place in our body. What is the role of gene? What is the role of RNA? What is the role of the protein synthesis? What is the role of final metabolism? Okay, so when we study about all these things, we study about the transcript of the RNA part. We are studying about the transcriptomics. Okay. It says requires high quality RNA sample, large scale data interpretation challenges, or all that. Provides insights into gene regulation, captures dynamic responses to environmental or dynamic changes. This all you can read. Okay. I think you understand now now proteomics. Now the third stage was, what was the third stage? Third stage was protein synthesis. So anything, the study that studies about that protein synthesis is proteomics, large-scale study of protein, including a structure, function, and interaction. Disease biomarker discovery, identify protein signatures of disease, drug development. Again, everything is around here is about understanding how things are working in our body, because if we know that, we can develop a suitable drug. Understanding cellular processes, as already said, investigating signaling pathways and protein networks. Providing functional insight before beyond genome and transcriptome. Yes. What is saying? We understood the genome, we understood the gene, what information it has, we understood the transcriptome, how that information is converted, and now we are understanding how that is converted into proteins. Okay, so this was proteomics. Now we are talking about epigenomics. What is epigenomics? Now epi means above genome. Now above genome means it understands complete set of epigenetic modifications in the genome. What it does mean is, look, the gene, that is our scientist has all the information, but there are certain codes, there are certain processes which regulate that gene expression. Means gene expression means how that information is transferred, how that information is read is the part of RNA, but before that also there are some epigenetic modifications which regulate gene expression. Okay. So this help in understanding cancer, diabetes, neurological disorders, and all that. So methods, these methods you will have to learn. UPSC can directly ask this: bisulfite sequencing or chromatin. What are these? Immunoprecipitation. What are these related to? Recently seen in news. Okay. Now next one is single nucleotide polymorphism. Now single nucleotide polymorphism, you need to understand what is single nucleotide polymorphism. Okay, yes, before this, I wanted to cover this one. Yes, I forgot it. Genetic markers in conservation biology. Genetic markers, to be very precise. Now, you all know we are all are humans, or let's say there is a population of tigers, there is a population of lions. But despite belonging to the same species, we all are humans, but we are different. Then why are we different? Because although 99% of our DNA is same, okay, 99% of our DNA, let's say in our country, India, all of us have same DNA, but there are some areas in our DNA which makes us different, okay, which makes us different. How it makes us different? Let's say there is a person who eats a lot of rice, okay, and he might get fat, but there can be another person who eats a lot of rice, but he doesn't get fat. Why? Because because of that difference, that 1% difference. Because our bodies are built differently, and these are known as genetic biomarkers, genetic markers, and these can be microsatellites. UPSC asked this question: what is microsatellite DNA? Okay, these can be single nucleotide polymorphisms. These can be mitochondrial DNA markers. Okay. So look, and this is the application: monitoring genetic diversity. Why there is diversity in our populations, identifying distinct populations, tracking illegal wildlife trade, restoring habitats. Because if we know about differences, what differences exist, we can understand, we can do multiple applications. Okay, up to applications I will come later, but let us first complete this SNPs. This is very important. Single nucleotide polymorphisms. This was in, I put this in the last year also. I was expecting this, but UPSC is going to ask someday. This, it seems difficult, but this is very easy. What is this? It simply says that after let's say there are 300 sequences. As you already know, we have AGCT, adenine, guanine, cytosine, and thymine. Okay, so this is how our DNA is formed, the cyclical structure, AGCT. But what happens is, after some numbers, there are some changes. Means, let's say I am X and there is one person Y. Sorry, I, X and Y, you can confuse. There is one person A, there is another person B. So X up to 300 sequences, 300 nucleotides, everything is same. But at the 301st nucleotide, what happens? There is a change. So this change is known as single nucleotide polymorphism. Are single base pair variations in DNA sequence among individuals occurring approximately every 300 nucleotides. Most SNPs are found in non-coding regions, but they have profound impact on regulatory of the coding regions. Okay. So why it is important? Again, if you have to create some personal medicines or for some person, you will have to understand their own gene for drug development and all those things. DNA microarrays, next generation sequencing, all these are methods, you can just learn them. Okay, now coming to next one, DNA fingerprinting. Again, this is like that only. DNA fingerprinting, what we do, we create again, as I told, 99% people of organisms of same species have same DNA. Okay, but 1% there is variation, and that 1% we use for DNA profiling to identifying individuals from a population based on their unique DNA sequences. So this can be done using microsatellites. Look, microsatellites UPSC has asked, short tandem repeats. If UPSC has asked microsatellites, then it can ask short tandem repeats also. Okay, short tandem repeats, microsatellites, these are something unique that we found uniquely in different individuals. Okay, so this is how they are used in DNA fingerprinting. So please read this, it becomes easy once you understand this. These are very important topic. Okay, horizontal gene transfer. Horizontal gene transfer, you need to understand, this is very, very important topic. UPSC has already asked DNA gene silencing in 2019. UPSC can ask horizontal gene transfer any year now. What is horizontal gene transfer? Normally what happens, I got my genes from my mother and father. They got from their mother and father, and they got from their mother and father. Okay, so there is normally a vertical gene transfer from offspring parent relation. Okay, but there is also a horizontal gene transfer. How it happens? Let's say a bacteria is interacting with the virus. Okay, and let's say means somehow bacteria gets some genes from virus. Okay, and that virus had gene of pathogenic resistance. Means pathogenic resistance means that virus had already developed resistance to some drugs, but this bacteria didn't have. But now this bacteria, since it interacted with that virus, it has now those genes, and those genes are the only thing that they need to develop that resistance. Okay, so transfer of genetic material outside traditional parent offspring inheritance, as I already said. Now how can it happen? Transfer, uptake from free DNA from environment, they can take up from environment. DNA transfer mediated by viruses, as I already said. Conjugation, DNA transfer of DNA between bacteria through physical contact, example plasmid exchange. Now let me give you a very, very interesting example. Let's say there is an organism, or let's say there is a bacteria. Okay, let's say there is an algae. Okay, and algae, you know, has photosynthetic ability, means algae can produce its own food. Okay, now let's say some microorganism interacts with that algae. Let me just say simply, though it is not that simple. Let's say that microorganism eats that algae, and somehow that horizontal gene transfer, the gene which is responsible for photosynthesis in algae, it is transferred to that organism. So that organism will become a super organism, means it will have its own characteristic, plus it will be able to do photosynthesis also. Okay, so that is the significance of horizontal gene transfer, and it is very important. Why? Because it can lead to widespread antimicrobial resistance, because those organisms which do not have antimicrobial resistance can get it from those organisms which already have those, because of horizontal gene transfer. Okay, so these I know were very difficult topics, but believe me, UPSC is asking this. And genome sequencing, DNA barcoding, DNA fingerprinting we read about. Okay, aerial metagenomics, bioinformatics, transcriptome, everything that I have discussed is asked by UPSC. Just watch the video again, you will understand it. If you do not understand it, I will make the video again in a more simpler manner. Okay, take care, goodbye.